Hi, this is Miss Litton, and this is the 7th period review with excellent AP Bio students. Say hi. Hi. Are you anxious?
Yes.
I know, I'm sorry. Just got to give those finals. Got to help hold you accountable. So let's do Unit 3 review. If we look at Unit 3, we did energy and enzymes, right? What kind of things come to mind when I say energy? It was just like big ticket items. ATP, I like that. What else? The sun has energy. Let's try to think. Yes? Photons, sun. Come on, peeps. Metabolism. Maybe some more. How about laws of thermodynamics? How about entropy? This, my friends, is what you have to do. You have to be able to see the big ticket. Some of you get so detailed. It's like you memorize a single leaf and you forget what forest you're in and what country you're in. Okay? You got to think about what are all the things related to energy. Bring it back to your big ideas. Okay? So, energy. Second word, enzymes. What are some things that come right to mind when we think hats and hoodies are off, when we think about enzymes? Substrate. What else? Active site. What else? Allosteric site. Cofactors. Do I hear... You can put it back up. Do I hear... I love you. I love you. Do I hear... Hashtag memes. I just was like, I've never seen that.
What can I do? My hair just folds up and comes
What? Okay, it's a good thing you're handsome. Okay, in a non-creepy teacher way. I even have a hard time keeping it together now. Okay, come back. I didn't hear energy of activation because that's what enzymes do. They lower it, right? They lower the energy of activation. That's why they make reactions more likely to go forward. Okay, we did a lab on enzyme activity. What? What was that lab? You used the enzyme catalase. What did you break down? Hydrogen peroxide into what? Oxygen and water. What did you measure? Oxygen in parts per trillion. What did you use? An oxygen center, where, right? What were your variables? Concentration of hydrogen peroxide. You could have done the temperature of the enzyme, right? Those are all different things we could have measured with that. Okay? Big ticket, photosynthesis. When I think photosynthesis, what comes to mind? What's big ticket here? Light dependent? Light independent? What else comes to mind? Chloroplasts, I like it. Pigments, I didn't hear what you said.
Oxygen.
Oxygen as a product, right? I would probably focus not so much on oxygen, but the idea that you're fixing what? No, fixing what? Carbon, right? Using the energy from the light-dependent reaction to synthesize sugar, okay? When you think about photosynthesis, did we do a lab on photosynthesis? Yes. What did we use? Spinach leaves, right? We punched them out. What did we do to those spinach leaves before we started our lab? We took out not just oxygen, we took out the... gas is in between the spaces of inside the leaf, right? And we filled that instead with what? Water. And the idea there is, what did we vary? What was our independent variable? Concentration of carbon dioxide by using various concentrations of baking soda, bicarbonate. And the idea would be if you increase the CO2, then you should increase the rate of photosynthesis because it has more what to work with? Substrate, reactant, right? So you have more CO2 to work with. And so you would expect to have, therefore, more product. What was our product in this case? Oxygen. Same product as before with our enzyme lab, right? Same product as before, but this time we measured it by how fast it made the leaf discs what? Float. Because they sunk because you made them more dense by filling their air spaces with water. that water happened to be infused with carbon dioxide. Some wasn't though, right? We did distilled water too, didn't we? And that would have been the control, right? So that control, that water, we wouldn't expect the leaf disc to rise or rise as quickly. Do you agree with me on that? Big ticket. All right, cellular respiration. When we think about cellular respiration, what are some big ticket items? ATP, use of glucose. But what are we doing in cellular respiration? What's the word we want to use there? Oxidizing. Thank you. We are oxidizing glucose so we can take the energy out of the bonds between the individual carbon atoms and use that to synthesize what? ATP. Using the intermediary who? NAD primarily and a little FAD. Okay? When we talked about cellular fitness, right, we said if you have a bunch of different molecules available to you, right, you could then adapt to different environments, yes? We talked about phospholipids having variations within them so you could withstand different environments, okay? We talked about the universalness of what? Glycolysis, because who does it? All cells. So that contributes to our idea of a common ancestor, yes? So remember to bring it back to those big ticket items. Okay? Those are all important. Okay? So in a little more detail, what are the laws of thermodynamics? Energy cannot be what? Created or destroyed. It can just be transferred. And every time you transfer, you lose some as heat. So it's not 100%, right? How does this relate to entropy and energy? This right here would be what? High or low entropy? High entropy. Does it have a lot of potential? No, but it is very what? Stable, because it can't get any worse. We applied those ideas of entropy to molecules. Who has more potential, glucose or CO2 and water? Glucose has more potential. Is it more stable? No. Does it have more or less entropy? Less entropy, right? But more potential energy. So when you oxidize glucose and break it down to CO2 and water, it gives off energy, which we ultimately use to make some ATP. And this process right here is called what? Cellular respiration. If we go the other way from CO2 and water to glucose, if it gives off energy this way, if this is high entropy, right? High entropy, low potential, we must have to put energy in it to make glucose, yes? Where does that energy come from? The sun. The big yellow one's the sun. Shines light, right? And we use that energy from the sun in order to take this low energy, low potential and make it higher potential energy. And that process is called what? Photosynthesis. Who can harvest the sun's energy? What in plants? Chlorophyll is one. Be more general. Pigments. Do we have only one pigment? No. This is an example of fitness of a plant because it has a variety of pigments, right? Cellular fitness. If you have a variety of fitness, you can absorb different wavelengths, right? In order to excite your electrons. So keep thinking big picture. Now this looks like to me like a gradient, a proton gradient, right? Where have we seen this? In both cellular respiration and photosynthesis, whenever what? What is that? Electron transport chain. In the electron transport chain, you are either using high energy electrons that probably came from water who are excited from the sun, or high energy electrons brought to an electron transport chain via reduced NAD or reduced FAD. You with me on that? Those high energy electrons are getting dropped off energetically, not physically, energetically at the high end of the electron transport chain. The electrons are passed, and as they are passed, that energy is captured. Is it 100% transferred to the next molecule? No. What law says no? Second law. But as it gets transferred, some of those coenzymes embedded in the membrane can get enough energy to take a hydrogen from one side of the membrane and put it on the other side of the membrane. And as you create that high concentration of this would be like water behind a dam. These are like people trying to get into Disneyland. As you have more of those hydrogen ions, they want to go to the other side. When they go to the other side, they will move through the same thing in cellular respiration and photosynthesis. What? Say my name. ATP synthase complex, and you synthesize ATP. Now, both photosynthesis and cellular respiration synthesize ATP. Photosynthesis is synthesizing just enough, actually a little bit over, every time it does the light reaction in order to have enough ATP and a little bit over in order to do the dark reaction, the Calvin cycle, the light independent reaction, right? But you'll notice plants don't just have a chloroplast because chloroplast can make ATP. Its job is to fix sugar, right? Because plants need sugar too. Or what do we call it? PJL, G3P. G3P is the building block of everything in the plant, right? And so that's why it does photosynthesis. It's taking the sun's energy and using the sun's energy to take fixed CO2 using RUBP, our gardening tool, using RUBP and energy from ATP and reduced NADP in order to synthesize G3P. That's the job of the chloroplast. It still needs a mighty who? Mitochondria. Because the mitochondria knows how to take that G3P, or glucose, right? Take the glucose. 2 G3Ps would be a glucose, but take that molecule and oxidize it, and now transfer that energy. It was sun's energy, then it was G3P energy. Now it's going to be transferred into ATP energy, which is the coinage of the cell. We good? Okay. Tell your bio buddies, do you have any aha moment there? Did you put anything together? Feel a little stronger? Okay, now come back to me. Come back. So we know that that catabolic reactions release energy and anabolic reactions require energy. And if we do them at the same time, what is it called? Coupled reaction. A catabolic reaction will release energy, and anabolic reaction requires energy. We looked at two categories of reactions. We looked at exergonic versus endergonic. Exergonic, the delta G goes what? Positive or negative in exergonic? Goes negative because the products have less energy than the reactants. Whereas in endergonic reaction, our delta G goes positive because the reactants have more energy than the products. Okay? Wait. What did I just say? Endergonic reaction, the products have more energy than the reactants. I don't know what I was saying, but you were all saying it with me. Okay, to make any reaction go faster, what could we use? Enzyme. Because you know you have to have energy to do an endergonic reaction, but exergonic reactions, there's a little hurdle there. And the reason is everything wants to break down. You would just fall apart, okay? But you're not falling apart because you have these little hurdles. We call that the what again? Energy of activation. What do enzymes do? Lower the hurdle to make the reaction more likely to go forward. They do that by binding with the substrate and facilitating some sort of change. It could be a building reaction, it could be a breaking reaction, okay? But they make that happen because they have a special area on the enzyme called the active site. Primarily enzymes are made out of what important molecule? Proteins. Those proteins have a special shape. The protein shape is ultimately dependent upon the primary structure. Oh, back to the DNA that coded for it. Yes, ma'am. Okay? But the primary structure, the sequence of amino acids in the protein, right? That's the primary structure. The secondary structure, we have two choices. What is it? Alpha helix, beta beta sheet. That is only due to the interaction between the carboxyl group and the amino group, yes? But the tertiary structure is the folding pattern. Chaperone proteins can fold those proteins in the right shape so the active site is there to bind to the substrate. If the active site is not the right shape, it will not bind to the substrate and the reaction will not go forward. That's not always a bad thing because sometimes you have enough of that. So sometimes you have something that binds to the active site to stop the actual substrate from binding to the active site, and we call that a competitive inhibitor. If you don't bind to the active site, you bind to a secondary site, what's that called? Allosteric. And that can still change the active site, right? Because if it's a protein, we know that folding pattern, it could be quaternary structure, more than one chain. If something binds on, it sets off a series of movements within that protein, which could ultimately change its shape. Okay, and that could be regulatory. So we know it lowers the energy of activation. We know things that affect enzyme activity, concentration of the substrate, concentration of the enzyme. But here, when the substrate increases, eventually it levels off. Why? Why does it level off? All the active sites are full, saturated. I love it. Temperature, pH can affect it. Inhibitors can affect it. You had essays relate the chemical structure of an enzyme to its specificity and catalytic ability. Did we just go over that? We did. Okay, design a quantitative experiment to investigate the influence of pH or temperature on the activity. Did we go over that? Yes. Describe what information concerning the structure of an enzyme could be inferred from your experiment. If your enzyme is altered by the temperature, the pH, right, a competitor, then it's not going to be able to do its job, right? And you expect your product to be less. In our case, oxygen that is released. Next, cellular respiration and photosynthesis. The equation is the same, just in reverse, right? What gets reduced, what gets oxidized, right? All of that, you want to be familiar with that. On cellular respiration, there's like kind of a big overview. We noticed here that it says oxidative phosphorylation. Why is that called what is happening here? When you look at this diagram, what is happening here? The ETC. Why is it called oxidative phosphorylation? You need oxygen at the end to capture the electrons. So you can keep removing the electrons, and when they do that, oxygen electrons binds with hydrogen, forms what? Water, right? So that gets oxidized, but also think about at the top of the chain. Who comes in at the top of the chain? NAD and FAD, and they are reduced at that point, right? They're reduced. They show up at the top of the electron transport chain. What do? They drop off their electrons, and they get what? Oxidized. Oxidative phosphorylation. Okay. Now, here, 2 ATP are made, but it doesn't say oxidative phosphorylation. What kind of phosphorylation is that? Substrate level phosphorylation, just a handoff. I have a phosphate, now I give the phosphate to you using an enzyme. Okay. We know if oxygen is present, this pyruvic acid is going to get converted into what? To acetyl-CoA, right? Then the acetyl-CoA are going to get fed into the Krebs cycle. Krebs cycle, another name for it, citric acid cycle. Then you do your nana-nana, your electron transport chain, so you can make a bunch of what? ATP. But if you don't have oxygen, you still only make 2 ATP. Why do we do fermentation if you don't have oxygen? Why do we need to do it? Oxidize the NAD, because when you do glycolysis, you've got to reduce some NAD and make some ATP too. So you've got to keep reducing your NAD. If all your NAD in your body is in its reduced state, you can't do glycolysis even, so you've got to oxidize it. Okay? And we learned the location of the different reactions. Glycolysis occurs in the cytoplasm. Transition prep, we move into the mitochondria. Citric acid cycle, Krebs cycle in the matrix of the mitochondria as well. The electron transport chain. These folds are called what? Cristae. So on those folds, that's where you're going to do your electron transport chain. Reductase, cytochrome oxidase. We're pushing hydrogens out into the inner membrane space. When they come back in through ATP synthase complex into the matrix, that's when you make some ATP. Okay? All right, now, there's a picture of the Krebs cycle, you know this. If you don't have oxygen, by the way, this entire presentation, the sequence of these pictures is in the stream, Google Classroom in the stream. Okay? Here, you're showing fermentation in your 2 byproducts. Either depends on the organism you are. You and I make, makes our muscle sore. Plants and yeast make alcohol. Either way, you oxidize your NAD. Perfect. Okay, questions on that? The net balance equation for cellular respiration is quite simple, but respiration is a complex process. Do you think I would expect you to know how to write out the equation of cellular respiration? Yes. Okay. Using diagrams, written explanations, explain it, blah, blah, blah. Describe a lab in which you could measure respiration. Did we do a lab where we measured cellular respiration? No. Am I going to hold you accountable to it? No. What do you think you could measure? It's got to be products or reactants, one or the other, right? If they ever gave you a lab, you could measure that. You could measure that. Okay? And I'm not going to ask you about it right now, but before your AP exam, I'm going to give you a lab about it because I know what I'm going to ask you. I'm not going to ask you a lab on that, but I will explain a lab to you just in the off chance you need one. Okay? Yes?
We need to know how to draw anything for the...
You always need to know how to draw things.
Okay.
All right. Next, photosynthesis. I know we did photosynthesis first, and this is the order I have. Okay, we know about chloroplasts, we know about the structures, we know about what occurs where, right? What occurs on the membranes, what occurs on, what are those membrane names, by the way? Thylakoid membrane. What occurs on them? The light-dependent reaction. What occurs out in the stroma? Light-independent reactions, good. And so this picture is here to review all of that. Here is showing you the different wavelengths of which you can absorb the different pigments. You're not overwhelmed. Take a breath. You know this dance, literally. Okay? And we can do it once if you would like to do that. Okay, we will. Okay. Here's another summary. Remember, the purpose of the light reaction is to give you reduced NADP and some ATP, so you can use that in the Calvin cycle, dark reaction, light independent reaction, right? What is a product of the light reaction that the plant does not need for photosynthesis? Oxygen. But what does it need oxygen for? Cellular respiration. Do you think a plant does more cellular respiration or more photosynthesis? What? Photosynthesis. How do I know this? Because you are alive. If a plant did not do more photosynthesis and cellular respiration, you would have nothing to breathe right now and nothing to eat. Right? Go obvious, people. Okay? We talked about that there is photosystem 1, photosystem 2. Photosystem 2 starts with what? Water. What's the formula for water? H2O. Photosystem 2. Right? Sweep the two hydrogens from H2O. Yeah? Okay? Then you use photosystem 2 to make your ATP. Photosystem 1, the same electrons as you had before, are excited, but this time we don't let them cool down and we reduce some NADP. Perfect. Then we're ready for the Calvin cycle. We start with RUBP and we bind it to who? Carbon. RUBP carboxylase or Rubisco is probably the most prolific enzyme on the planet because that is the enzyme that binds CO2 to RUBP. Makes sense that that would be the most prolific, right? Okay, and then we go through these series of reactions by which we invest energy in reduced NADP, electrons from NADP, and we end up with 6 G3Ps or PGALs. We have all 6. Do we cash them out now? No, we do not cash in our chips. We only take 1, and we leave the remaining 5 so they can be reorganized so you can keep doing this dance over and over and over again. Yes? Okay, perfect. Now, there's a problem with RUBP though, right? Yes, there's a problem with RUBP because on hot days she closes her stomata and doesn't do as she oughta, right? And instead of hooking up with who? Instead of hooking up with carbon dioxide, she hooks up with for shame. Okay? That process is called what? Photorespiration. And photorespiration is not good for a plant. It's not photosynthesis, it's not respiration. # no. Okay? So there are 2 adaptations that some plants have to deal with this. One is called C4 plants. They have a molecule, so here's bad. Okay? C4 plants have and CAM plants both have something called PEP. PEP is loyal. PEP will only hook up with who? CO2. So why in the first place would CO2 levels run low where I even need PEP? It's hot. I'm closing my stomata. I'm still doing photosynthesis. So what gas am I consuming out of that space? Carbon dioxide, and I'm producing a lot of So CO2 levels go down, O2 levels go up because I don't have access to more free air. Yes? That's why. So PEP will hook up with CO2 and escort CO2 into the dark reaction, which is isolated in the bundle sheath cells that are surrounded by mesophyll cells with Krebs anatomy. Okay, it looks like a wreath. Basically, you're isolating your dark reaction. Remember the wreath? Okay. Secondary way, second adaptation is turn. Did I push record? I did. The second thing you can do is, can plants fix CO2 at night? When it's cool outside, they're not worried about water loss. They fix it, and then if they need it during the daytime. Does fixing the CO2 to PEP cost you? Yes. But the benefits must outweigh the cost, okay? And so, net balanced equation for photosynthesis, explain the steps. There are the steps, describe a lab. Okay, we already went over that lab, yeah? Okay, you want to sing? Yeah. And after we sing, I have a hint for you. Would you like to have a hint? Yes. Everybody loves hints. Okay, so let's start with cellular respiration. What's the whole thing called? Cellular respiration. First step. Don't move slowly. Let's get this done. And what's it called? Glycolysis. Who does it?
All cells.
Where?
In the cytoplasm.
What do you start out with? Glucose. How many carbons?
6.
What are you going to do?
Break in half.
Steps.
10.
Enzymes.
10.
Called 2
pyruvic acids.
And you reduce some NAD and make some ATP. 2. Oh, we have plenty of oxygen. What should we do? Aerobic respiration. What's the next step called? Transition prep. Here we go. And you reduce some NAD and 2 acetyl-CoA, put 1 away. 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP, reduce NAD, and 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP, reduce NAD, and stop. Gather all your reduced NADs and FADs and take them to the top of the ETC. Reductase, cytochrome oxidase, and you make some ATP. A lot of it, or a lot of it. We catch electrons at the bottom. Oxygen forming water. Water. What are we going to do? Break it in half. What do we get? Oxygen and electrons. Sweet. Here comes the sun, traveling in waves, particles of light are called photons, and it gets the electrons excited. PU, and you make some ATP. Here comes the sun, traveling in waves, particles of light are called photons, and it gets the electrons excited. Who catches it? NADP, forming reduced NADP. Then you take some ATP and some reduced NADP to the dark reaction, or the Calvin cycle, or the light-independent reaction. 3C5 plus 3C1 makes 3C6, break down to 6C3. Use some ATP, use some reduced NADP on your 6C3. Take 1 away, PJL, G3P. And you're left with 5C3. Use some ATP to build 3C5 plus 3C1 makes 3C6, break down to 6C3. Use some ATP, use some reduced NADP on your 6C3. Take 1 away, glucose. And you're left with 5C3. Use some ATP to build 3C5. Stop. Pick up your glucose. Break it in half. How many steps?
10.
Enzymes. 10. Called
2 pyruvic acids.
And you
reduce some NAD and make some ATP. 2.
Oh no! We don't have the oxygen. What are we going to do? Fermentation. How many choices do we have? 2. You and I make lactic acid.
Makes our muscles sore.
Plants and yeast make alcohol. Either way, you oxidize your NAD. Yay! Okay. Questions that you have? Oh, you want the hint? You know what I worry about? What's a disease? Yeah, I do worry about you. I pray for you every day. Do you know what's a very sad disease that spreads? Very bad, besides stress. Bad disease you know about, terrible thing, people get it, they have to go see a doctor. Why? Where's the flu? What? What?
HIV.
No, not cancer. Cancer makes me sad. Cancer makes me sad. What's your hint? Cancer makes me sad. That's your hint. Yeah. All right. What?
What about cancer?
You tell me. You will find out about it. You've already learned it. Okay. Okay. What else? Do you have any other questions? What? No, I gave you so many hints. I've given you like 6 hints for 10 essays.
I think one more.
Listen to me. Listen to me. Shh. Show some fortitude and some character. You're sounding weak right now. I don't like people begging. When you study tonight, you cannot sit there and think, I'm going to read every chapter. It does you no good to stay up till 3 in the morning. It will not do any good, right? You know the different items that we have talked about. You know the different essays. You know what I'm going to ask you for sure. Know those, right? We went over that in class. Know them for sure. Okay? Do the best you can. You know what? Go for a walk with a friend and talk biology if you want to. Have something good to eat. Don't eat junk tonight. Please eat healthy. Please eat good for you food. And go to sleep. If you can't sleep and you have anxiety, say, okay, I'm going to get up and study for a little bit, but get off your screen because your screen's going to make you stay awake. So read some notes or something that you printed out. Look at your lab notebook or something like that. That would be a really good thing to do. So you're off your screen so you can go to sleep. Get up, have some good breakfast, think positive thoughts, and believe in yourself because I believe in you. Yes?
For the free response, if we were to write about like the structure of like the periodic table and like atoms and like atomic mass and stuff, could we get full points?
Sounds kind of small. That sounds kind of small. And you would only write about it if I never talked about it. If I never talked about it. Yes?
Is it only that the question can't be on this essay final, or can it?
Yeah, it's whatever, yeah. If I ask you anything related to it on the questions I ask you in class, you can't make that be your free choice.
So if it was on a past test, that's okay.
Oh, yeah, totally. I don't expect you to memorize what was on, every test. Like, no, I asked that in Unit 3, Version 2. Okay, no. What else? You done? Okay, go study. Yes, Luke. Wait, I can't hear. Shh, go.
For the independent thing, whatever you want, like you said, that seemed too small. So that would that mean like if you didn't talk about like photosynthesis, you would talk about all of photosynthesis?
I would tell me a lot about it. Yeah.
Okay.
Yeah, I want to know that you're a master of a topic, not a memorizer of about 4 or 5 facts.
So you could just talk.
Yeah, when I see memorizer of 4 or 5 facts, I'm like, 5 out of 10.
So you could just talk about like light.
Wait, say that again?
So you could just like talk about the light reaction, not be all of photosynthesis or something.
Yeah, you're trying to convince me in that essay. Don't shoot for the minimum, right? That's weak. You're better than that. Shoot for telling me, convincing me that you know a whole bunch about this topic. But also think about this. We have the whole class period, but every single question is worth the same amount of points. So if you spend 40% of your time writing on one question, I'd say hashtag poor choice, right? You need to learn how to control it. Like, look at your time and go, okay, we have this many minutes. Like, let's do the math right now. If we start in every class 10 minutes after, that's when the timer starts, right? Then that means you have 60, 70, 80 minutes, right? Okay? So minimum maximum you would have 8 minutes per question to respond. And ideally you'd want to get it down to 7 minutes per question because you want to have enough minutes to proof it at the end to make sure you're okay, right? So get in the mind because that's the same mindset you have to have for the College Board. When time's up, time's up. So you want to be able to go. The upside is I'm not having you write in complete sentences. You can write in bullets, but you need to support your statements that you make. Okay? And some questions will be like super fast. You'll be able to do it in 3 minutes, and so you'll invest more on another question. But you're going to keep in mind, you're like, okay, 10 questions. After 5 questions, what's 5 times 7? 35 minutes in, it would be a great idea to be done with 5 questions, right? Pre-prepare for your time so you're not in a situation where you're like, when can I have more time? I have 2 questions left. Because the answer is going to be no. Okay? No, because that's what you're going to have to learn how to do for the AP exam. Okay? Anything else you want to ask me? Okay? Yes? Go ahead.
Do we have any bonus points on this test?
No. You don't. Your bonus is that I gave you 5 of the essays. That's your bonus.
6.
Appreciate that. 6. That's right. I gave you a hint. Yes?
Do you have any other helpful study tips?
I just think you should sleep. I do. You need to bring your best brain tomorrow. You may laugh, but it is when you are not anxious, right? When you are calm, cool, and collected and thoughtful, you're more likely to do a good job. People who are under stress make poor choices, right? So just be like, I've got this. Keep telling yourself, I've got this. It'll come to me. It'll come to me. Just don't spin around. If you approach one and you just can't think, stop, go to another question, let your brain work on it in the background. What you don't want to just do is spin around the drain, spin around the drain, spin around the drain, and not move forward. It's better that you do X, right? Because you'll still get an A on this test if you do 9 questions well and you don't even do 1 question. You still have an A, right? Still have an A on the essay.
Assuming that you did well.
Well, I am. That's what I said. Assuming that you did well on the other 9 questions. Yes?
Is the essay and multiple choice going as 2 subjects?
No. No, 1 assignment. 100 and 100 makes 200. It's double a test. What else? Anything else? Yes, I'm sorry. I don't do it as percentage. Points are points. I'm a big, so go look how many points there are. But every minute you spend calculating what you need to get on the minimum grade to get the grade that you want in the class is a wasted minute. It makes no difference. 0 difference. Because either you'll go, hey, I can get a 70 on this test and still get an A, and then you'll screw up and you won't get it. Okay? Or you're thinking, did I do enough, right? It doesn't matter. Do the very best you can on every single thing. It does not matter. Do the very best you can. But you maximize what you can do, right? Surround yourself, right? Tonight you're not going to break up with your boyfriend or your girlfriend. Hashtag no breakups. Hashtag no parental fights. I don't care if your parents look at you and say, you are the worst thing on the planet. You say to them, thank you, Mom. They would never do that, but you would say, thank you, Mom, thank you, Dad, I love you too, I'm going to study. Keep away from your mom of your own. I love you. Make good choices.